My dad can eat a mountain of french fries with bacon and cheese.
Yeah, well, my dad can eat 5 buckets of fried chicken.
My dad can eat 1,000 chicken nuggets.
My dad can eat a quadruple burger.
My dad can eat 500 corn dogs.
Yeah, well, one day I'll be able to eat twice that.
With obesity becoming a new epidemic, there has been an increasing amount of research linking obesity to diseases and the shortening of lifespan. One of the most popular areas of research was actually on the underlying mechanisms between caloric restrictions and lifespan extension. According to many of the researchers available, there are actually 2 general mechanisms for how SIR restriction extends lifespan. But I will be focusing on the regulated response, which is B, which involves the use of the protein SIR2. Now, let's take a look at how aging occurs. In yeast cells, there are ribosomal DNA repeats, and when there is a replication defect, a circular copy of the ribosomal DNA pops up by homologous recombination to form these extra-chromosomal rDNA circles known as ERCs. And an accumulation of these ERCs are actually found to be one of the factors of aging. But the presence of SIR2 actually has the ability to inhibit the RNA recombination by silencing the chromatin and thus extending the lifespan. So, how does chromatin silencing occur? SIR2 is a histone deacetylase, and it requires NAD as a co-substrate. In an open chromatin, the lysine present in the amino terminal tails of histones is acetylated, while in a closed chromatin, the lysine is deacetylated. It is actually the SIR2 that is stimulated to carry out this reaction in the presence of NAD, in which the available levels of NAD are coupled to the metabolic rate of cells. Now, what do you mean by metabolic rate? This will be explained later. So, both the functional SIR2 and NAD need to be present in order for the chromatin to be silenced and the lifespan to be extended. This lies in the role of NAD as a mediator of caloric restriction. In excess calorie individual A, glucose is oxidized at a high rate by glycolytic enzymes during Krebs cycle in the mitochondria. This uses up a portion of the available NAD from the common pool, and this would mean that the SIR2 activity will be relatively low. But in individual B, where there is caloric restriction, the flow of carbon through glycolysis and the Krebs cycle is low, thus increasing the amount of available NAD and elevating the SIR2 promoted silencing, therefore increasing lifespan. In conclusion, the extension in lifespan by caloric restriction is a regulated response requiring SIR2 and NAD. This regulation involves a qualitative shift in metabolism that can be sensed by SIR2. The deacetylase activity of the enzyme SIR2 must then slow any degenerative processes that limit lifespan.